trial presentations. Your grade's all full. This one, nothing terrible will happen to you if you don't remember a single word. Unlike all the other presentations you had. I was in Berlin a year ago. I was giving this poster, and I noticed that nobody was stopping by my poster. As a matter of fact, this woman walked by, looked at my title, and jerked her head away. And so I sort of socially tackled her. I didn't physically tackle her. I sort of socially tackled her. I said, "Oh, did you just recoil from my poster?" And she said, "I'm sorry. As soon as I see genetics, I do this. I close my eyes, shut my ears, and I keep on walking." Because she didn't want to be like infected with genetic stuff, right? So I thought, you know, there are some things that are happening, and I thought people should know about. Things that are happening. I have no disclosures. Nobody tries to private geneticist, you know. So you remember the central dogma: DNA goes to RNA, RNA goes to proteins, and there are lots and lots of DNA in your body. Only 1 to 2% of which actually codes for proteins. So you were probably taught that all the rest of it's just junk. Those introns, junk doesn't do anything. Turns out that's not actually true. The other thing was the number of genes we had. So back in the day, the first sequence was of a fruit fly. They had 13,000 genes. And they sequenced something called a roundworm, 19,000 genes, and then the mustard weed. So we figured that humans had 200,000 genes. That was the estimate, 200 to 400,000 genes. Then we actually sequenced the human genome, major accomplishment scientifically, and it turns out we only have 30 to 40,000 genes. Like, well, not as complicated as we did, but we're still winning, right? We're still winning. We're still beating the stupid mustard plant. Until 3 years later, when we sequenced Rna sativa, the rice plant in China, and it turns out that it had 56,000 genes. It's like, whoa, how in the world is this rice plant more complicated than my wife, right? It makes no sense whatsoever. How is that possible? Well, humans gain complexity through regulation and splicing, which is all that other part of the genome. So what we once thought was just junk, introns and junk, it turns out 70% of that is actually important to regulation, splice site variation, and creating the complexity that you have. And one of the things that we do as humans is we take one gene, and in some organs, we splice together certain numbers, say 1, 2, 3, and 4 exons, to use in the thyroid. We use the exact same gene in your brain, but instead we use 1, 2, 3, and 5, and it does the gene then does something completely different by adding on that extra exon. So we gain complexity by splice site variation and regulation variation across our body. Now, isn't this interesting? So all the endocrine people are studying this gene in thyroid. All the brain people are studying this gene in the brain, but there could be one variant that's causing both syndromes to occur. This is really challenging what we think about as far as disease and phenotypes in a way that we've never really understood before. What's a disease and what's a syndrome? A disease is a disorder of structure or function within the body. But a syndrome is a group of symptoms that consistently occurs together. We call this a syndrome. This really challenged, is Parkinson's disease a disease or is it a syndrome? Is it a collection of things that we find that goes together? Bradykinesia, resting tremor, rigidity. Or is there some common pathophysiologic event that in some patients presents with Lewy body disease and substantia nigra causing Parkinson's disease, but in other patients causes cortical disease, causing diffuse Lewy body disease, progressive supranuclear palsy, that there might be one common event that spreads out to these other phenotypic variations. As a matter of fact, is that even true with just one disease tree? For example, could there be overlap in neurodegeneration? That there's one particular susceptibility leading some patients to lead to tauopathies and Alzheimer's disease, but in other patients leading to alpha-synucleinopathies leading to Parkinson's disease. So people started to look for it, and they did find a lot of overlap between Alzheimer's and Parkinson's disease with specific genome-wide association studies for the key variants that were occurring in both conditions. So the BrainStem Consortium, just in June of 2018, decided to look at all this incredible genome-wide association study that we have to see if there are some commonalities between different phenotypes that we don't think of as being associated with each other. It took 200,000 patients, 25 brain disorders, 17 phenotypes, and in June 2018 they published. This is just a listing of the sample size of each of the variations, but This is really the heat map that shows the common variation that occurs. So anxiety disorders with major depression, that kind of worked out pretty well. So in psychiatry, that all worked. Then when they did the neurological diseases, some interesting findings: focal epilepsy and intracerebral hemorrhage may have a lot of genetic overlap to one another. That's kind of a curious one. Here's a little proof positive that's working: migraine with aura, and so forth. And then one of the more interesting things was when they took the psychiatric diseases and they crossed it with neurological diseases, and they found things like migraine association with epilepsy. I'm sorry, focal epilepsy. This is still back to the brain one. Focal epilepsy with intracellular hemorrhage, attention deficit disorder that matched up with depression, schizophrenia with almost all the other psychiatric illnesses. And then they looked across at neurologic and psychiatry overlap. And one of the more interesting things for me was they found this high genetic overlap between anxiety disorders and intracellular hemorrhage. So there, theoretically, could you have some sort of epinephrine stress-related hormone that makes somebody anxious, but also increases your risk of brain hemorrhage? And there was, in fact, analysis that we did that found that stress was an independent risk factor for intracellular hemorrhage. Schizophrenia with ischemic stroke, where the heck did that one come out from? And yet that was one of the most highly statistically significant findings throughout the study. So these kinds of phenotypic variations from what common genetic mechanism, anxiety with epilepsy, stress and epilepsy is another field of research that's out there, is changing the way we think about the phenotype. What is the phenotype that we're actually studying? Because all we see is the end product, the end conditions that we are now able to define clinically by looking at someone. Here's another fun one: chronic traumatic encephalopathy. Your brain gets concussed and concussed and concussed, and eventually you concuss it enough that it develops this progressive neurodegeneration, a progressive process, not a one-time injury that does it. It continues to advance and advance, causing more and more brain injury. And Gehrig's disease, so there's a theory that chronic trauma may lead to a particular triggering event that in some patients leads to chronic traumatic encephalopathy, but maybe in others leads to Alzheimer's disease, and another person may lead to ALS. And if these common elements are occurring together from a common mechanism, maybe we shouldn't just be studying this. Maybe we should be studying the collection of things to see if there's common variation. So these new phenotypes. So one of the things that people are doing right now is looking at trauma and injury and looking for common things. And one of the things, of course, is an inflammatory response that can occur after trauma to see if they can find new targets that may manifest in such conditions. One of the newer techniques that are out there is called RNA sequencing. This is whole transcriptome screening. So just like we had whole genome screening, now we can do whole transcriptome screening, all of your mRNA. But this doesn't include the mRNA, it also includes something called microRNAs. These are little regulators, they don't actually code for proteins. Methylation, gene silencing. As we age, there's gene silencing changes. In some patients, that leads to cancers, and others leads to neurodegeneration. Your DNA looks exactly the same. There's no variation in it. It's just whether it's methylated or not. It predicts whether or not you get the disease. So we can identify those genes that are abnormally transcribed or expressed. Again, there's nothing wrong with the gene sequence. It's just abnormally transcribed. So this was a paper on TBI and genome-wide transcriptomics, and this is just an example. If you ever see one of these, it looks really scary. Just go to the red. The red is the highly significant key regulator of all of the blue that's around it. So these are the two proteins here that was abnormally expressed in those that ended up having disease versus those that didn't have disease. So to summarize that section, what we understand about specific phenotypes may be changing in the future as we discover new pathophysiologic mechanisms. What I've been studying is deep intracellular hemorrhage may have huge overlap with leukoariosis, which may have huge overlap with vascular cognitive hemorrhage. There may be some one single defining thing that leads to all three of those conditions. So the next thing I want to describe very briefly is something called linkage. The closer two places are on the chromosome, the more likely they are to be inherited together. And I've created a Danu special drawing. Let's pretend there's a gene here for, say, stroke. And that's what the yellow bar is, but we don't know where that one is. But we know exactly where these other markers are, say A and B. Because B is physically closer to the exact gene that causes stroke, for example, it will be inherited with it much more often than A will. A is just too physically far away. So when we do these genome-wide association studies, we find something significant. We're not saying that thing is the disease-causing gene. We're saying it's inherited with the actual gene so often, it's way more than by chance alone. That's the concept of all these genome-wide association studies. So in the GWAS study, we look at hundreds of thousands to 1 million markers across the genome. This gives us 90 to 95% coverage of your genome. And because of linkage, we have great coverage. And then we look for association with disease. It could be protective or it could be risk. And again, most of the hits that we're finding, surprisingly, are in these gene deserts where there aren't any exons. We thought, well, I thought this thing did nothing. And it turns out it does do something. 70% of your genome is associated with either regulatory, microRNAs, and all these other features that we thought didn't do anything. If you use a p-value less than 0.5 that's scientifically valid for significant association, but you're looking at 1 million markers, then by chance alone, we'd have 50,000 positive hits. So we do essentially a Bonferroni. We correct for 1 million markers. And so what we consider to be statistically significant is 5e to the minus 8, which is a really, really tiny level. And for that, you need massive sample sizes. So that's what the last 20 years we've done since sequencing the human genome. We've gathered these massive sample size of subjects, genotyped them all. This is the 2012, 6 years ago, human map of all genome-wide significant hits across your chromosomes. It's an enormous amount of work. You have to understand, back in 2002, this thing had 0 genome-wide significant hits on it. Now, this is the fill-out that's occurring from all these genetic studies. You probably missed this, but back in October, in the fire hose of news that is the modern era, this little report came out. A new gene therapy could literally end a fatal brain disease. And in the New York Times, in the first gene therapy halts a fatal brain disease. Then it was published in the New England Journal of Medicine, and it had to do with ALD. This is the old Lorenzo's Oil movie, if you, for those of us who are as old as I am. And it's an X-linked adrenoleukodystrophy. It usually affects boys typically younger than 12 years of age with a progressive white matter disease. They eventually become more and more responsive, and they die. Universally, within a decade, actually the vast majority within 5 years of being diagnosed with the condition. Transplant was the only effective therapy that must be begun before the person ever develops any neurological disease. We identified the protein, ABCD1, many decades ago. We figured this out. The vast majority of people have a spontaneous mutation. You can't look back in your history and say, oh, we have a family history of this, we should look out for it. That's actually not the way the vast majority occur. The vast majority are spontaneous. And it leads to accumulation of very long-chain fatty acids. So you may have not have heard of lentivirus, but you've heard of the most famous lentivirus, which is the HIV virus. So these crazy scientists took the HIV virus, which has a particular receptor for your immune system, and they removed the HIV part of the virus, the part that kills you, but they reprogrammed it with a normal ABCD1 gene. Then they infected the cells of the immune system of these patients. They first wiped out their bone marrow, then they infected their cells with this modified HIV virus. They carried the normal ABCD1 gene. And because these immune cells also penetrate the blood-brain barrier, go into your brain, it also infected microglial cells within their brain. Sorry, I'm going to skip through some of this for time. So they use apheresis to gather patients' immune cells and they transplant them. So this is the results of the vectors that have showed up in these patients. Drug product infusion, and at 6 months, it was still persisting. The vast majority of patients, they were still producing this product. And here there is months from infusion, and they continue to produce the normal product, the normal gene within their cells. And these are some of the patients. 10 out of 12 kids treated were essentially cured of their ALD. So this was this was the boy that was initially diagnosed with ALD that led to the diagnosis in this boy. This was the boy that was diagnosed with a disease that led to the diagnosis in their sibling. And the siblings were treated in the condition. 10 out of 12 have essentially gone years and still show no signs of the disease itself. It's miraculous, right? Now, it was technically an ideal scenario. The cells of interest were thankfully blood cells. The virus which targeted these was a reverse transcriptase, which affects blood cells. And they didn't have to require the removal of the abnormal DNA, you just had to produce the normal DNA, and the patients would get better. But wouldn't it be great if we sent just the right DNA piece, so that part of the brain, and fix that part? So I need to tell you about this particular bacteria called Streptococcus Thermophilus. It's used in the dairy industry essentially to create yogurt. And for whatever reason, we discovered that these bacteria have an enormously selective immune system, better than humans have. They can defeat almost any kind of virus that's thrown at them, and you can't get really a virus from a lot of these bacteria. This is the actual device that my wife uses to make yogurt. And the first time your spouse cultures something with bacteria and hands it to you, you're thinking, I don't want to die, I don't want to keep this thing up. And, my wife made from yogurt this yogurt that I ate and I survived. And I thought to myself, well, I won't die because it has CRISPR-Cas9 in it. So these yogurts have this system called CRISPR in it. For every cell in the world, there are 10 viruses. That's how many viruses there are in the world. And yet these bacteria have developed a way that not only they, but all their subsequent generations can defeat these viruses. It's called CRISPR-Cas9. What they do is when one bacteria survives a particular virus, they snip out a critical piece of its DNA and they store it in their own DNA. Then when they transcribe, that gets carried on to all their future generations so that their future generations know how to find this particular virus. It transcribes this thing, and these are like the guys at the airport carrying those little plaques. You know, this is Lori Gutman virus on it, right? And they're wandering around the side of us. Are you Lori Gutman? Are you Lori Gutman? And when it finds Lori Gutman, it says, hey, I found her. And hey, Cas9 come over. Cas9 goes over and kills Lori Gutman. It kills that virus, it snips that double-stranded DNA. That's what this CRISPR system does, just wanders around doing this. This is a not as fun way of presenting that. Anyway, so somebody said, wow, that's really cool. I wonder what would happen if we put a human DNA between these CRISPR things. So they did. They put human DNA in it, and sure enough, it bound to human DNA, and it snipped it. Cool, right? But the curious thing that happened next was that your own body's mechanism fixed it. Our DNA repair mechanism then took a strand of DNA and transcribed it in there because it didn't want to leave this gap in your DNA. So we thought, hey, what if we could select what gets transcribed into this place where this system has snipped? We could find a specific place in the genome, snip it out, and then transcribe something in that. This is basically how it works. It looks for a particular sequence, and then it makes a guide RNA to that. Cas9 sees it, snips it, and then your body, we can tell it exactly what to transcribe in this place. So in 2012, we were able to do it. Edit any double-strand DNA part in a cell with CRISPR's technology. This was published in Science in 2012. And this is just the showing of it. In 2015, this got sophisticated up to the point that they were able to shorten trinucleotide repeats. So they took DNA that had over 300 trinucleotide repeats, and they were able to reduce it 200 trinucleotide repeats. Think about myotonic dystrophy, Huntington's disease, Friedreich's ataxia, being able to remove those trinucleotide repeats. You don't have to come up with a drug to make them better. You don't have to come up with some other conditions, so forth. Just remove those trinucleotide repeats. It can make a huge impact in these patients' lives. In 2017, scientists were able to CRISPR in that normal dystrophin protein, normal dystrophin gene, into muscle cells that were producing the abnormal dystrophin gene, and those muscles recovered. One of my favorite ones is this one. So ApoE4 is the most prevalent genetic risk factor for Alzheimer's disease. And just in May of this year, May 31st, 2018, scientists were able to convert ApoE3 cells into bad ApoE4 cells using CRISPR. Then they converted them back. So they took the ApoE4 and converted them back to ApoE3 cells. Can you imagine what could happen if we could CRISPR out ApoE4 and put in a normal ApoE3 into patients who are suffering from disease? This is the demonstration of the difference in amyloid uptake in those with the ApoE4 and then those that were CRISPRed out back to ApoE3 in these cells. So CRISPR technology has really reached a tipping point. This is the publication related to CRISPR and how it's taking off. You're going to hear more and more about this. Just remember the yogurt thing. the viruses and so forth. And gene therapy is coming a long way with other things. This was a story of a 1-year-old with leukemia, refractory to all the treatments, was waiting for a bone marrow transplant. They extracted T cells from a healthy donor. They deactivated the immune genes in it using a CRISPR technology, then reinfused the cells back into the person. So this wasn't a donor that matched her. This was just any donor. And they were able to block out their immune cells so that she could survive until a transplant could be found. And it worked. She had this, she waited long enough, and then she had a more permanent transplant that occurred. In 2016, in Nature, they removed immune cells from recipients, disabled a particular gene for the immune response to cancer. They cultured it and they reinfused it back into the patient. The patient survived. So this led to the first human trial in human technology. The NIH sponsored a trial looking at this particular gene, knocking it out in particular cells in patients who have metastatic small cell cancer. There are 15 participants. It started in August of 2016. We hope to hear the results in the near future. Keep your eyes out. The first real CRISPR human trial. They take peripheral blood lymphocytes, they collect them, they program them, and then they send them back into the body to try and kill this specific cancer. Just a little bit ago, July 11, 2018, one of the problems is trying to get the cells to accept the CRISPR. But this technique that was published just in July used what's called electroporation. So far, we don't actually just CRISPR your body. We take a cell out, we CRISPR it, and then we send it back into your body. But that process is harder than you would think. So this group of investigators were able to take the cells out, use this electroporation. They got 95% of the cells to accept the CRISPR, the change in the DNA. Sorry, I go a little faster. However, there is what I call the oops factor. So unfortunately, the system isn't perfect. There's actually sometimes accidental editing of other parts of your genome. We're all going along, so happy to think that if you use this 20 nucleotide sequence, it must be unique in your body. But it turns out when you really look, it turns out that CRISPR is putting the cassette into other places within the genome as well. So there may be errors that occur through the process. So there's another technique called Rna Interference. And in this, the Rna Interference is a complementary strand to the diseased RNA. And it just comes along and binds it, essentially inactivates it. It's, as a pharmacologic tool, it has almost no side effects to the RNA. You infuse the RNA and it binds just a set amount. It doesn't stay with your system forever, and it just binds out that particular sequence temporarily. So this was used in lung transplant patients. In 2012, there were lung transplant patients who were getting respiratory syncytial virus infections, which leads to a marked amount of disability. They did a randomized, double-blind, placebo-controlled trial with an aerosolized small RNA inhibitor just to the RSV that prevented it from replicating. The study reduced the incidence of progressive bronchiolitis obliterans from 50% down to 6.3% by using the RNA inhibitor therapy. So it makes an inhibitor to the thing the RSV needs in order to replicate. And so it's amazing what people are doing with these RNAs. PCSK9, there are drugs that can affect this particular site, but we also are able to create RNA inhibitor to this particular gene, and thereby lower patients' cholesterol. Once you're done with particular treatment, you can stop giving them the RNA inhibitor, and it just goes away. So investigators are searching for ways of using inhibitor RNA for other kinds of viruses, hepatitis B cure someday, the common flu, cancer treatment, doing targeted cancer therapy, treatment of heart failure. There's a patent to use RNA inhibitor to to silence presenilin, also to block oncogenes before someone gets cancer. Once you find out somebody has a risk, to block the expression of those abnormal genes. Then the final thing I want to talk about is pharmacological biology. There are many failed drugs for one indication or other that have a specific target within the human body. I want to tell you a story of a 39-year-old. I was at Grand Rounds in Harvard when I heard this story. There's a patient with a 39-year-old who pretended in a coma, required emergency decompression for their craniopharyngeal genome. 7 months later, they came back in a coma, they had to be decompressed again. Again at 8.5 months, and at 9 months, and 9.25 months. Every time they resected, it grew back and caused the patient to fall into a coma, and they had to have it resected again. Now, they have to present to a hospital. The investigators had just finished reporting in 2016 that this thing called BRAF V600E was the cause of nearly all craniopharyngiomas. So, went online, Googled BRAF inhibitors. Oh, look, there are already BRAF inhibitors out there for the treatment of like melanoma and other kinds of conditions modestly effective. So they did something I consider a little crazy. They gave it to the patient. So this is the MRI on day 0 of the craniopharyngioma. They started this BRAF inhibitor based upon nothing more than knowing that that was something that caused the aspirated craniopharyngiomas and that the drug existed. And they reduced the tumor over 90% in size. And they continue on therapy. They added, it worked so well, they added a second therapy on top of it. This is incredible. And they're actually doing a phase 2 trial for all craniopharyngiomas with this drug target based upon that. So genome sequencing is now possible for roughly $1,000, what used to be the cost of an MRI. That's how cheap it is to do a whole genome sequence these days. So they started doing that in metastases. They took out brain mets, compared it to the lung mets and breast cancer mets. And they found that 53% of brain mets were actually susceptible to some already FDA-approved drug that was available. So the idea here is that in the future, we'll be able to take someone's cancer and truly do personalized medicine, find out exactly what drugs will affect this particular cancer the most, and give that to patients. If you try to study any one of these, it may work for 5% or 10%, and it will be failed drugs, the effect size wasn't big enough, 90% of patients died. But if you were but for a certain group, it may affect 90% of their tumor, not just 5%. So in summary, gene therapy is coming. And here's the things that you need to know about. There are viral therapies that can be used to affect the correct cell type. That's what we have to figure out next, is how to gene edit in the body, not just take the cells out and edit it. There are also a multitude of things that we use RNA inhibitors for, and CRISPR to affect things in the germline. One of the things that people want to do is for Huntington's and other trinucleotide repeats to get fetuses or embryos to find the cell, CRISPR out all the trinucleotide repeats, and then that's the only thing they got to cut out, and see if we can prevent the disease from occurring. Geneticists understand genetics really well. Neuroscientists understand neurology really well. What I implore you to do is, when you have the opportunity, try and bridge that gap. Some of these therapies, some things that they're discovering really could affect a lot of clinical trials in the future. And this is my key. Thank you.
For a dead woman, you really book, you know? So, one of the things from a trials perspective that's always bothered me about the gene therapy trials is that they promise home runs, right? I mean, the effect sizes that are expected, if you get just exactly the right thing, it's either going to cure the person or not. Is that a realistic expectation for our trial designs? I mean, should we design the trial so that if it doesn't save 75% of the people that get enrolled, it's a negative trial and we should walk away from it? Or should we be looking for modest size things and do much larger trials like we would for drugs?
Right. These are molecular geneticists. They're not clinical trialists. That always drives me crazy when I see an N of 6 paper that gets into Nature. Right? It's like, well, I don't know, it's just a small N, the sample size, reproducibility. I see so many grants that come in that don't even have a power sample size calculation on them because, I don't know, that's the way they do things. Definitely, you're absolutely right. I was shocked that 10 out of 12 of the kids in the ALD trial responded to the therapy. That was a home run. But yes, I expect a lot of things in the future not to have that kind of home run. How do you do it without they have to go through the exact same process we do for pharmacology, which is to test it, see what the estimated effect size we think there's going to be, and try to move forward from there. But without other knowledge, they're just doing the best they can on that, trying to figure figure out how much do we think this is going to work. What happened to those 2 kids that we did all the exact same things to, but for whatever reason they didn't get cured from the ILD, they still developed disease. They worked, by the way. They definitely just transcribed the correct protein, but they still ended up with disease. So you're absolutely right. As geneticists working in a lab, when we do something, it affects usually the entire petri dish, right? But in humans, it's going to be a much more complex thing.
I just looked up those 2 kids who did not respond. One of them had withdrawn and was pre-symptomatic before the trial, and the second one was withdrawn just for an unknown reason and died. So actually, they did not actually get the event.
If you had stayed with the therapy. It's not the easiest therapy, right? The wipe out your bone marrow, they take your cells out and give them back. But what's amazing was that the effect lasted, that they were still producing the protein years later. That's really amazing. All right, thank you very much.